guys, welcome back to my channel. Today we'll do enzymes again. I will speak about the mechanisms of regulation of the enzyme activity. So some enzymes with specialized regulatory functions respond to allosteric effectors or covalent modification, or they can also show altered rates of enzyme synthesis or degradation when physiological conditions are changed. So first we'll speak about allosteric enzymes. So they're regulated by molecules called effectors. They're also called modifiers, so the same thing. They bind non-covalently at a site other than the active site. So these enzymes are usually composed of multiple subunits, and the regulatory site, also known as the allosteric site, binds the effector. So the presence of an allosteric effector can alter the affinity of the enzyme for its substrate, or it can also modify the maximal catalytic activity of the enzyme, or both. Effectors that inhibit the enzyme activity are termed negative effectors, whereas those that increase the enzyme activity are called positive effectors. These enzymes, as I've mentioned in my previous video, show a sigmoidal curve when the reaction velocity is plotted against the substrate concentration. For example, the glycolytic enzyme phosphofructokinase 1 is allosterically inhibited by citrate, which is not a substrate for the enzyme. Moving on to covalent modification. Most frequently, it is modified by the addition or the removal of a phosphate group or groups from specific serine, threonine, or tyrosine residues of the enzyme. These are amino acids. So there are two ways in which it can covalently modify an enzyme. So one is phosphorylation and dephosphorylation. Now, phosphorylation reactions, they are catalyzed by a family of enzymes called protein kinases that use adenosine triphosphate, which is ATP, as a phosphate donor. Phosphate groups are cleaved from phosphorylated enzymes by the action of phosphoprotein phosphatases. So phosphorylation is by protein kinases, dephosphorylation is by phosphoprotein phosphatases. Now, depending on the specific enzyme, the phosphorylated form may be more or less active than the unphosphorylated enzyme. For example, phosphorylation of glycogen phosphorylase, which is an enzyme that degrades glycogen, increases the activity of the enzyme. But the addition of phosphate to glycogen synthase, an enzyme that synthesizes glycogen, decreases the activity of the enzyme. So phosphorylation can either increase or decrease activities. The second method is induction or repression of enzyme activity. Now, for example, elevated levels of insulin, which is a hormone, as a result of high blood glucose levels, cause an increase in the synthesis of key enzymes involved in glucose metabolism. So because of the increase in the level of one hormone, there is an increase in the regulation of one enzyme. So the difference is that alterations in enzyme levels as a result of induction or repression of protein synthesis are slow. They take hours to days, but compared with allosterically or covalently regulated changes, which occur in seconds to minutes. You can see that all the regulator events are immediate except for synthesis or degradation of the enzyme, which takes hours to days. And you need to know about the results, the change in Vmax and Km of allosteric and covalent modification. But there is only a change in the concentration of the enzyme with induction or repression. Now we will move on to the last part of our video, which is Zymogens. They're also called proenzymes. So they are inactive precursors of the enzymes. In general, Zymogens contain extra amino acids in their sequences compared to the actual enzyme. This prevents them from being catalytically active. So some examples are procolipase, which is activated by trypsin in your intestine to colipase. So procolipase is the zymogen and colipase is the active enzyme. Then you have pepsinogen in your stomach converted to pepsin, and then trypsinogen to trypsin. That's it for this video, guys. Hope you enjoyed it. See you in the next one. Bye.